Hello guys and welcome back to Learning with Isha. In the last video, we have already spoken about a process called as glycolysis. We have already seen that glycolysis in simple words is glucose breakdown. Now, glucose is a 6-carbon compound. So, this 6-carbon glucose is broken down by these glycolysis reactions into two molecules of pyruvate. So, as you would expect, a 6-carbon compound will be broken down into two 3-carbon compounds, that is, two molecules of pyruvate. The story we are going to look now is what happens with pyruvate in the cell. As I have already told you previously कि जो ग्लूकोज ब्रेकडाउन अप टिल पायरुवेट का मैकेनिज्म था वो सेम था both in the case of aerobic respiration and anaerobic respiration. So whether oxygen is present or absent, the mechanism is going to be the same. But after here, the story changes drastically. So we are basically going to look at what could be the different things that happen with pyruvate in a cell. With this introduction, let's move on and look one by one at what happens with pyruvate. To understand that, let's consider one molecule of pyruvate. As we have already discussed, this pyruvate is a 3-carbon compound. So let's try to draw the structure first. Let's draw a carbon backbone. And here we have CH3, here we have COO, and here we have CO, double bond O and one O minus. This is your COO minus, this is the carboxyl group. So this is the structure of your pyruvate. Now what happens with this pyruvate? Number one, this pyruvate might be converted to lactic acid. In the second case, it might be converted to ethanol and carbon dioxide. And in the third case, this is converted to acetyl-coenzyme A and carbon dioxide. So you see, this 3-carbon compound is giving me different products in different cases. Let's look at what are the scenarios that are leading to the differences in the products. In the first case, lactic acid is being formed. This represents an anaerobic condition. Anaerobic condition means the condition having absence of oxygen. Now, here this lactic acid is also a 3-carbon compound. Why? Because you see no CO2, nothing is evolved. So all the 3 carbons are conserved here. Where is it observed? It is observed in the case of some yeasts, some bacteria, and very commonly in the case of our muscles. Now if you see, if you're not used to exercising and suddenly you decide to run 3 miles, you'd realize that you've got a sudden cramp in your leg. पैर में होता है ना दर्द झंझनाहट. Exactly. Why is that? It is due to the accumulation of lactic acid in your muscles. Why? You are not used to running that much. Your muscles are not used to taking up that much oxygen. So in the absence of oxygen, in the deficiency of oxygen, anaerobic conditions are established in the muscle. In this anaerobic condition, instead of forming acetyl-coenzyme A, lactic acid जमा हो जाता है हमारे muscles में. Nothing to worry about when you rest or you put your feet in hot water. Again, this lactic acid is quickly broken down and we get relief from the pain. So this process is known as lactic acid fermentation. Why is this known as lactic acid fermentation? Because lactic acid is being formed. Okay. In the second case, we see ethanol is being formed. As you would expect, this is known as ethanol or alcoholic fermentation. Why? Because ethanol is actually an alcohol. So this is known as ethanol or alcoholic fermentation. This also happens under anaerobic conditions. Anaerobic conditions, guys, I hope you're aware. It means conditions that are oxygen deficient or actually oxygen absent. So in this condition, oxygen is actually absent. So you will just write absence of oxygen in both of these cases. Now you see ethanol is a 2-carbon compound. How did I come to this conclusion? Because 1 carbon is here in the carbon dioxide. So even if I did not know the structure, I could have made कि 3 carbon से 1 carbon यहाँ आ गया तो 2 ही बचा ethanol में. Anyways, in the third case, acetyl-coenzyme A and carbon dioxide is formed. Since two conditions are anaerobic, let's have some aerobic conditions as well. So this is the aerobic conditions. And after this acetyl-coenzyme A would go on into the Krebs cycle, and then there would be phosphorylation and things that we are going to look at in the subsequent videos. Let's talk about each of them a little bit more in detail. Lactic acid fermentation, we know lactic acid is being formed. Which is the enzyme involved in this case? The enzyme involved is lactate dehydrogenase. This lactate dehydrogenase enzyme does what? See, this is the structure of pyruvate. What is going to be the modification in the structure of lactic acid? See, this is going to be the same. So you will have COO minus. Instead of this ketone group, here I am going to have an alcohol group. So OHH and the bottom structure, the methyl group, is going to remain the same. This is the structure of lactic acid. You see, from here to here, from ketone to alcohol, the process is of reduction. Why? Because reduction is the addition of hydrogen. So we know कि हमारे biological systems में कभी भी reduction अकेले नहीं होता है. It is coupled with oxidation. Why? Because in biological systems we always have redox reactions, right? So अगर ये reduce हो रहा है तो which is the molecule that must be oxidized? That is in fact NADH plus H plus is converted to NAD plus. So these hydrogen groups will be utilized here in the reduction process. How much ATP will be generated here? Only 2 molecules of ATP are generated here. Therefore, we say that anaerobic respiration generates very less amount of energy. So this was the first type. Now let's go ahead and also look at what happens in the ethanol or the alcoholic fermentation. When we talk about alcoholic fermentation, this is a very commercially important fermentation. Whatever brewing you are doing, right? If you're making any alcohol or if you're making any bread, all these fermentations in fact involve this alcoholic fermentation. So as you might have guessed, हमारा जो yeast है favorite Saccharomyces cerevisiae, जिसे हम bakers या brewers yeast कहते हैं, वहाँ पे ये बहुत ही commonly observe होता है. So where is it observed? It is observed in the case of yeast and many other bacteria as well. I was telling you the commercially most important example. Now if I talk about what happens here in terms of structures, it isn't going to be very difficult if we check this out step by step. So let's just begin with the structure of our pyruvate molecule. First, make the carbon chain. It's a 3-carbon compound. नीचे आप CH3 डाल दीजिए. यहाँ पे क्या होगा? Ketone group हो जाएगा. And definitely it's an acid. So you're going to add COO minus group. As simple as that. Now this pyruvate molecule is converted to a 2-carbon compound. This is 3-carbon to a 2-carbon compound. This 2-carbon compound is acetaldehyde. Okay, so acetaldehyde means there is going to be an aldehyde group and 2 carbons. Let's draw a 2 carbon chain. Okay, now bottom में आपका CH3 ही रहेगा, ऊपर क्या आ जाएगा, एक एल्डिहाइड ग्रुप. सो कीटोन की जगह क्या आ जाएगा, एल्डिहाइड ग्रुप. Double bond O and just add one H. CHO is what? Aldehyde. So this is acid aldehyde. Now you see 3 carbon to 2 carbon, we are losing one molecule of carbon. Where is it going? It must be evolved as carbon dioxide, right? Now we know all biological reactions have catalysts known as enzymes. So the enzyme involved here is going to be pyruvate decarboxylase. Pyruvate का decarboxylation हो रहा है. What is decarboxylation? It is removal of carbon dioxide. So the enzyme here will be pyruvate decarboxylase. And in fact, wherever you see decarboxylase, you will definitely see that a CO2 is being lost, being evolved, being removed. Okay. Now at this step, what more do we require? Many enzymes require something that we know as cofactors. These cofactors allow the enzyme to work better. A cofactor in this case is thymine pyrophosphate, TPP. Besides, also magnesium ions. So this one is optional, but the enzyme name is a must for you. You must remember, okay? So we have reached a 2 carbon compound known as acetaldehyde. Now from acetaldehyde, we are going to go to another 2 carbon compound, which is going to be an alcohol. Which alcohol? Ethanol. So here CH3 common. Here you're supposed to add an alcohol. So OH डाल दो. And 4 valency है, so we need to add 2 hydrogens. One here, one here. So basically CH2OH and CH3. What is this? This is ethanol. So we have reached ethanol now. Now here 2 carbon to 2 carbon, so no need of any carbon removal, no carbon dioxide. What could be the enzyme involved here? Well, the enzyme over here is an enzyme called alcohol dehydrogenase. Well, what is the meaning of this name? You see carefully. What is being formed? Ethanol. Ethanol is what? It is alcohol, right? Now, what do you mean by dehydrogenase? It is a dehydrogenation reaction. Why? Because you see, here it is a ketone group that is being reduced to what? Form an alcohol group. So, this is a reduction reaction or dehydrogenation reaction. Now, you see here, if there is a reduction, there definitely has to be an oxidation also. Because in biological systems, reduction अकेले कभी भी नहीं होता है. Oxidation के साथ होता है a redox reaction. Reduction, oxidation. What is the oxidation involved here? Now you see the NADH plus H plus is going to get converted to NAD. Now this step is going to give me hydrogens that I am going to utilize over here. So this is a redox reaction. Now let's just move on quickly and come to the last kind of reaction where pyruvate is going to get converted to acetyl-coenzyme A. Now we've come to the last fate of pyruvate, that is the conversion of pyruvate to acetyl-coenzyme A. As we have already discussed, this is a very crucial molecule and it is finally the molecule that is going to go in Krebs cycle. Please take note of this fact that the previous 2 steps were taking place in the cytoplasm of the cell and oxidative decarboxylation will take place in the mitochondrial matrix. So please note that the location is the matrix of the mitochondria. So this is an important point over here. This step leads to the conversion of pyruvate to acetyl-coenzyme A. Now what we need to note here is that there are a series of reactions that are going to cause it. Now, all these reactions are catalyzed by PDC. What is PDC? It is the pyruvate dehydrogenase complex. Now, pyruvate dehydrogenase complex consists of three enzymes E1, E2 and E3. The three enzymes E1, E2 and E3 stand for pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. तो ये तीन एंजाइम्स हैं जो कि हमारे पूरे रिएक्शन को कैटलाइज करने वाली है. Please take note that even if you don't remember the names of the enzymes, do remember that the pyruvate dehydrogenase complex has three enzymes in it. Okay. Now also you need to remember that there are 5 important cofactors that will help these enzymes perform better. What are they? Now let's go sequentially. The first one is thymine pyrophosphate as in TPP. The second one is lipoamide. The third one is going to be FAD. The fourth one is going to be NAD. So as in flavin, adenine dinucleotide and nicotinamide adenine dinucleotide. Okay. Now the next one is your coenzyme A. Now when I write coenzyme A, this SH represents the thiol group. And as we are going to look at in a moment, this thiol group is going to be very critical in this reaction. So let's go step by step. Let's come to our good old pyruvate molecule. So this is my pyruvate. This is a 3-carbon compound, right? And we know acetyl-coenzyme A is a 2-carbon compound. So definitely there is a loss of carbon dioxide. Where is it lost? It is lost in fact at the first step. The involved enzyme is E1, pyruvate dehydrogenase. Now what happens? Now this part is gone. This is lost. The remaining part is going to get attached with TPP. Now again in the next step, this is going to get transferred to lipoamide and the TPP is going to be regenerated, right? Now again the lipoamide is further going to transfer it and the lipoamide is again going to be regenerated. Where is it being transferred? It is going to be transferred to coenzyme A. This reaction is extremely crucial. Please see what is happening. Now you see this coenzyme A has this thiol group. इसका सल्फर है. इससे क्या अटैच हो जाने वाला है? ये वाला ग्रुप. So you have COO and CH3. So now please be careful. This is acetyl and this is coenzyme A. So what have you formed? You have reached to acetyl coenzyme A. This is our molecule of interest. This is a 2-carbon compound. So sorry, 2-carbon compound. Okay, now you saw 1 carbon was lost as carbon dioxide and we reach acetyl coenzyme A. In this reaction, please also note what is happening here. FAD plus is getting reduced to FADH2. Now, if there is a reduction, we know there's going to be an oxidation as well. So now this is again going to get oxidized and NAD plus is going to get reduced to form NADH plus H plus. Now this molecule is what it is at energy currency. It is pretty much like a check that is not going to be availed right now but encashed later. We have already discussed in our video on ATP that ATP is instant energy. It's like cash. You pay it and you get whatever you like, right? Check, you have to wait. You have to let it go to the bank, then wo procedure hoga and you will get your money, right? Just like that, this needs to go through the electron transport chain to finally be able to give some amount of energy. Now, this entire reaction is known as oxidative decarboxylation. Let's just take a minute and write the net reaction for this process. So let's write the net reaction quickly. So I take pyruvate, This pyruvate is directly getting converted into acetyl-coenzyme A, two carbon compound, right? And one molecule of carbon dioxide. So one carbon here, so 3 carbon is satisfied. Next we saw NAD plus getting converted to NADH plus H plus. Also we saw that to form acetyl coenzyme A, we required what? We required coenzyme A. Just for a quick revision, I would tell you that here the enzyme involved is going to be the pyruvate dehydrogenase complex made up of E1, E2 and E3. And besides that, we had our 5 cofactors, TPP, and then we had lipoamide, and then we had NAD, and then we had FAD, and then we had our coenzyme A. So these were my 5 cofactors. You need to remember this, right? You need to remember the name of this complex at least and the 5 cofactors. This complex, all these enzymes also require an ion, that is our magnesium ion, right? And this process happens in the mitochondrial matrix. Well, that's all for today's video. I hope you enjoyed this video. This video serves as a basic for what? If you need to study fermentation, let it be lactic acid. Let it be alcoholic. You need to know this. If you need to study Krebs cycle, you need to know oxidative decarboxylation. So the next video is going to talk about nothing other than the Krebs cycle. We have already reached the mitochondria. Now let's see where the Krebs cycle takes place. If you have any doubts related to this video, feel free to contact me. Let me know if you have any suggestions for more such videos. If you need to know the details about lactic acid and alcoholic fermentation, do write in the comment section below. Don't forget to subscribe to my channel and share this video with your friends. Don't also forget to like the video. See you in the next video. Take care.